everybody, it's Mrs. Mancuso. Want to do another AP Bio review with you today. I would like to do cyclins. So cyclins literally has cycle right in it. So cyclins got their name because they tend to go up and down and up and down. They cycle within a cell. So here's a cell's normal life. Cells can either be in G1, or they can be in S, or they can be in G2, or they can be undergoing mitosis. And if you think about your cells, like here's my hand. The majority of cells in my hand, they are not in any of these steps right now. They actually are in what's called G0. So there's a checkpoint right here. And that checkpoint is to figure out what's my level of cyclins. Have I been making cyclins? So during all of this normal part of my cell life, I can make cyclins. This could be my cyclin level. And if my cyclin level goes up like this, I'm going to pass the checkpoint. And what that means is I'm going to go through S, which means I'm going to take every bit of DNA and I'm going to replicate it. I'm going to make 2 copies. I'm going to make identical copies because I want to make 2 cells, and both of those cells are going to need the DNA. So S is the replication of DNA. That involves helicase and opening up the bases and a leading strand and a lagging strand and really making an exact copy of my DNA so that I'm getting ready for mitosis. Well, in my hand, I'm not making cyclins. This is not happening, so I go into G0. G0 is when my cells are arrested. That means they are not in a stage of growth. They are not growing. I can stay in this for a long time, and if I need to, I can go back. If my cyclin levels go up, I can go back and divide. Like if I get a cut on my hand, I got to replace those skin cells. So I could go back, but a lot of times my body cells, because I am not in a growth stage of my life cycle, I'm going to stay in G0. All right, let's talk about a cell that's actually dividing. Let's talk about in an embryo. I've got to go from a zygote all the way to a baby. I've got to increase in the number of cells drastically, literally go up by billions. So I need to be doing this pathway. So I make enough cyclins. Cyclins are a very specific molecule. So let's say this is my cyclin. A cyclin bonds to a kinase. It's called a cyclin-dependent kinase. And remember, each different kinase within a cell is going to be specific in shape. And when it binds to this, these CDKs are activated. If you have enough activated CDKs at this point, you will begin this part of the cycle. What's really important is once you've passed a checkpoint, you need to degrade these and inactivate these. If you do not degrade it and inactivate it, when you come back around to pass the checkpoint again, it's going to be like an obvious pass. If this still exists, the levels are high enough and it's going to go again. And that's what happens with cancer cells. The cyclins don't get degraded. Well, it happens sometimes in cancer cells. If the cyclins don't get degraded, they actually pass the checkpoint where they really shouldn't. So you got to have degrading of your cyclins after each checkpoint. All right, the next checkpoint is over here. And this is a different cyclin. So I'm going to call it an M cyclin. And that cyclin is meant to go into mitosis. So my M cyclin has a different shape, and it binds to another kinase, and together they're called an MPF, maturation promoting factor. I'm testing the level of my MPF. If it's high enough, I'm going to pass this checkpoint. If I pass the checkpoint, hey, that means signal transduction pathway. Let's get mitosis going. What do I need to start mitosis? I need to take all of my chromosomes and start condensing them. I need to make my spindle fibers start to form from my centrioles. My centrioles have to go to the 2 opposite sides of the cell. What am I thinking about? All the beginning steps of mitosis. What do I need to do mitosis? So I want to go through prophase, metaphase, but before I do anaphase, there's a last checkpoint. The final checkpoint has nothing to do with cyclins. It has to do with spindle fibers. So here's my cell. Here are my chromosomes lined up at the equator. Here are my spindle fibers. There is a little pulling on the spindle fiber. And the idea is make sure that it's actually attached to the chromatid so that the chromatids will separate from each other. So a spindle fiber attaches to a centromere at the kinetochore region. This last checkpoint is making sure that that attachment has been made so that when I pull, I end up with 2 cells that each have one of those chromatids in it. If instead this was not all the way connected, I would end up with two cells. This one would have one of the early strands, this one would have one of the early strands, but then these two would still be connected and this would have nothing. So when chromosomes fail to separate, it's called non-disjunction. And non-disjunction could occur if this checkpoint is passed and it really shouldn't have. We know that non-disjunction does occur in the case of Down syndrome. The egg ends up with an extra of chromosome number 21. So the egg would actually have 24 chromosomes instead of 23, and the sperm would have 23 chromosomes. So someone with Down syndrome actually has 47 instead of 46 chromosomes because they have an extra of chromosome number 21. All right, so that is cyclins, and remember this is a signal transduction pathway, as most things are, that allow you to regulate when should you divide cells and when should you not. Thanks for joining me.